Welcome back for our final two back-to-back sessions for today's ICDA plenary meeting. We're starting with our second plenary presentation session, Mechanisms in Medicine. This session will be moderated by Charles Rotimi, who is an NIH Distinguished Investigator at the National Institutes of Health in the US and a founder of the Trans Institute Center for Research on Genomics and Global Health and the H3 Africa program, and is also a member of ICDA's organizing committee. Thank you so much, Charles, for moderating today.
Yeah, thank you very much. I, again, moderating this plenary 2, Mechanisms and Medicine. We have 3 speakers, and our first speaker is Xin Jin, a fellow at Harvard and the Broad Institute. She's going to talk to us about a top sequence system for in vivo investigation of functional. And she has done this work using autism. And she has a PhD in neuroscience from the Rockefeller University and a BS in chemistry from MIT. Take it away, Jane.
Thank you, Charles, for the nice introduction, and I want to thank the organizers for putting together this wonderful meeting. It's a challenging time, and I hope everyone is doing well. Thank you for taking the time to come today, and it's an honor for me to be here to share our work. Just like many of you, we're interested in developing a systematic approach to study gene function in a context that's biologically relevant, such as an intact tissue. Just like this cartoon demonstrated, we want to take these gene variants to understand the cellular and molecular substrate that they're operating on. And we want to understand what kind of cell type and subtype that they're operating on, the gene network that they are affecting, as well as in the future, their cellular neighborhood environments that's changing associated with these risk gene perturbations. And the tissue that I care deeply about is the brain, which is comprised of diverse cell types made during development to allow cognitions and behaviors. But when these development goes away, it results in devastating disorders such as autism. In the past almost 2 decades, fortunately, we have learned a great deal about these genes strongly implicated in autism. For example, a syndromic cases of autism called Rett syndrome is caused by the mutation in the single gene named MECP2. And MECP2 is a transcription repressor and activator that is required for both development and in the adult stage. But for a different autism risk gene, in this case, for example, SHANK3, it is a structural protein located at the synapse, which is the junction of 2 neurons, and it regulates neuron-neuron communication, among other things. So I could go on and on with these single genes that are already strongly implicated in autism. These single gene studies were very important because they really build the foundational models to allow the electrophysiology, molecular biology, and even behavior studies to approach this very complex question of autism. But compared to other diseases and disorders, our understanding of neurological disorders is still at a very infant phase. For example, we simply do not know when and where many of these risk genes act. And we also don't know how many different kinds or types of autism there are. So when we started this project, our question is, are these hundreds of risk genes affecting hundreds of different molecular pathways in the brain? Or is it possible that these risk genes can converge into subtypes that actually can be grouped for future mechanistic studies or even therapeutic considerations? Many of these genes express very broadly in development, so we wonder whether some of these risk genes can actually affect the same neuronal cell type or even affecting the same molecular pathways within these cell types. We want to take a genetic approach to study the loss of function of these risk genes and study how they impact a developing brain. And earlier today, we learned some great deal about the complexity of the genetic instruments in many diseases, and we will hear more about it tomorrow. But here for autism, we really decided to focus on a subset of these de novo variants that are simply protein truncation or loss of function variants. because these are likely to be highly penetrant with large effect sizes. We hope that this will serve as an important entry point to the complex autism biology. To study the gene function in cortical development and in the scalable way to study these long list of autism risk gene mutations, we stood on the shoulder of the giants who built in vitro perturb-seq. So in this case, in the context of cortical development, we can introduce these mutations in the risk gene that we care about through introducing the guide RNA injected lentivirus in the lateral ventricle in an early embryonic age at embryonic day 12. And this is such an early stage of corticogenesis that you can imagine some subsets of these neuronal stem cells will be labeled by lentiviral transduction and therefore confer the expression of the fluorophore as well as the CRISPR loss of function. In the subsequent 2 weeks of in vivo and in utero development, all the cellular progenies that are coming out from these infected neuroepithelia cells will carry out the expression of the fluorophore and the loss of function. So at postnatal day 7, our chosen time of analysis, we can enrich for these blue perturbed cells and use single-cell RNA sequencing to read out effect. And the single-cell resolution in here is absolutely crucial, not only because it provides a very high resolution readout of the mutation effect, but also importantly, allow us to actually multiplex this approach. So instead of injecting one vector at a time to study one risk gene at a time, we can inject a pool of lentiviral targeting a pool of 35 autism risk genes and only computationally resolve each individual cell and their perturbation identity. So by performing this in vivo pool genetic screen, our question is, across this cohort of autism risk gene perturbation, what are the molecular substrates and cell types that's affected by maybe convergent subsets of these autism risk genes? I want to say that this approach is scalable and therefore is obviously more efficient because we can study the mutation of many genes at once. But precisely because of this, our approach is more well controlled because it really allows us to compare the perturbation effect with the internally negative control vector that was injected in the same brain in the same day that was sequenced in the same batch. And this approach allows us to read out more robustly the perturbation effect because we can build a model to regress again any confounds such as the developmental timing and sequencing batch. I also want to highlight that there are specific features that comes with this experiment in our design. First, obviously, we are analyzing the effect out of the in vivo CRISPR experiments, and therefore this is a mixture of homozygous and heterozygous loss of function. We use 2 guide RNA on the same lentiviral vector for each of the risk gene to ensure the completeness of the loss of function. Also, what we're analyzing is actually a big mosaic brain that each of these clones of perturbed cells are sitting in the sea of wild type cells. And therefore, this approach allows us to focus on the cell autonomous effect instead of the sort of extrinsic non-autonomous effect. And ultimately, in this first experiment, we focus on the context of mouse brain development at a time window between embryonic day 12 to postnatal day 7. But in the future, we can expand this paradigm to analyze the perturbation effect in different developmental stages, such as later developmental stages that mainly synaptogenesis are happening. In the future, we can also imagine this kind of approach to be used in any other model organism and to study different tissue types. So here in total in this study, we analyzed the effect of 35 risk gene perturbations out of 17 independent experimental batches. So from the single cell analysis, we can classify these cells into 5 major cardinal cell types that's annotated by well-established markers thanks to many in the fields. We can identify the perturbation effect not only at the cardinal cell type level, but at a very high resolution. And this is because we can take any of these cardinal cell type and further classify them into subtypes. For example, for projection neuron here, we can take the projection neuron and further subtype them into several subtypes. Each of the subtype correspond to their laminar layer in the cortex. And from this data set, we can use several approaches, such as the machine learning-based topic modeling approach, to extract gene programs from these single cell data. Some of these gene programs seems to describe a specific subtype in the data sets, such as the top gene program and showing you here, the expression of which really correspond to a specific subtype of the excitatory neuron corresponding to layer 4 laminar position. However, we also observe a different kind of gene module, such as the one I'm showing here in the bottom. And you can see that this gene program expression is shared among many different subtypes of projection neurons. And this makes sense because this particular gene module describes a biological process of neurite development, which is a feature that's shared regardless of the laminar position of the projection neurons. So from this data set, we extracted a total of 14 different gene modules from 5 different cardinal cell types, what I'm showing you here by each of the row on the y-axis. And on the x-axis is each of the 35 autism risk gene perturbation. The size of the dots reflects the statistical significance. And even just by this like super bird eye view of the phenotypic landscape, we can probably appreciate that there are several levels of convergence even when we are analyzing this rather small pool of 35 risk gene perturbations. We observe that several modules are recurrently affected, shared by different autism risk gene perturbation. Some of these effects are quite cell type specific, such as the perturbation of this gene called ADMP affect a very specific cell type projection neuron in the projection neuron 1 module. And there are some other perturbations such as SCN2A that affect not only projection neuron as well as astrocytes. And this is really demonstrating the power of single cell analysis to capture phenotype that could be masked by the bulk sequencing analysis. Lastly, an interesting note to me as a neuroscientist, I set up this experiment and thinking of I will be able to discover neuronal phenotype by these perturbations. However, in our data, it does seem to that at this early developmental stages, both neurons and glia are directly affected by several of these autism risk gene perturbation. And I think this is particularly informative when we think about in the future to do in vitro modelings, often in the system with limited cellular heterogeneity. And this data set provide a guidance of what kind of cell type at what developmental ages to focus on in order to better model the disorders. So this is really a very rich data set. We have a lot of things to focus on. To follow up, we validated several of these results through orthogonal experimental systems. And in the rest of the talk, I will briefly tell you an example of a validation in the germline mutant rodent model about a glia phenotype. And this is a phenotype about a cell type called oligodendrocytes. It's a really fun glia cell type that can deposit myelin onto the axon of neurons to modulate the conductance as well as the connectivities of neurons. But at these rather early developmental stages in postnatal day 7, most of the oligodendrocyte cells in our data sets are actually still in the progenitor's stage, labeled here in the t-SNE plots. And we identified one of the gene module called ODC1, and the expression level of gene module ODC1 is quite high in the progenitor cell type, which represents that this might be a module that signals immatureness. We found out that the perturbation of 2 risk genes results in the decrease of this module. And one of the risk genes is called CHDA, a chromatin modifier that can regulate Wnt signaling by recruiting histone proteins. So independently, when we just saw this data set, there was a paper came out showing that using ChIP-seq experiment, the CHDA combines crucial genes that regulate oligodendrocyte differentiation and maturation, which builds confidence in our own data. So we went in and performed several validations. I'm showing you one of the experiments where we try to use in situ hybridization to identify oligodendrocyte progenitor marker expression, which is a module that we identify through the gene network analysis. We found out that in the postnatal day 7 mouse brain, in this heterozygous CHDA mutant model, just like what we discovered from the Perturb-seq, the progenitor marker in oligodendrocytes show about 30% decrease of expression, and effect size is similar to the Perturb-seq. So now we know that the maturation and differentiation is affected in oligodendrocytes, but there are really just more to learn, such as how these differences in the differentiation may lead to the physiological and behavior function of phenotype. This really leads to an interesting thought or hypothesis that these glial cells can operate through the non-cell autonomous effect that can modulate neural conductivity and circuit refinement in very subtle ways. And in the future, we're interested in looking into whether this oligodendrocyte phenotype persists in adulthood and whether this affects their roles in myelination in different brain regions and potentially affecting specific neuronal cell types. So to summarize, I told you an experimental approach that we built to think about a systems genetic approach in order to understand psychiatric disorders. And we hope that this will be a generalizable approach as a scalable genetic profiling method. And in the future, the perturbation side can be modified into CRISPR-A or CRISPR-I or other kind of genetic perturbation methods. And we can also imagine applying this kind of method in any other organs or animal model or in organisms to understand the mechanism of different diseases. And through this analysis, the single-cell data set really unveiled interesting biology that we think would have been masked by bulk sequencing represented by all these cell type-specific effects. And many of these were interesting follow-up. We're still learning from this data set ourselves every day. And we discovered that both neurons, particularly some synaptic gene modules, as well as glial cells can be direct target of several of these autism risk genes. And in the future, I'm really excited about the functional follow-ups that I demonstrated a very short example that the difference of mutant in differentiation of oligodendrocytes may influence the network. So starting from a pool screen in vivo, we identify a rare cell type at this point of development that may have been overlooked using the bulk sequencing approaches, and we validated this using the germline rodent animal. And I think this is really a very exciting time because these risk genes are important for clinical and biomedical reasons, but I also think they represent key entry point to fund biology because genes and molecules themselves don't give rise to thoughts and behavior. It is really the nervous system that does. So using approaches such as Perturb-seq allow us to take these genes and variants to understand what subsets of the nervous system that they act on to exert function or dysfunction. And briefly, in the future, we're interested in continuing using this functional genetic approach to understand the brain. And two things I didn't quite tell you is that the differences between mouse and human brain is a very obvious point that humans have some cell types that simply do not exist in mouse. And also by dissociating these cells for single cell analysis, I actually lost the spatial resolution of where these cells are and what these cellular neighborhoods are. And we set up the Perturb-seq to address these two questions by using the human cerebral organoid to study the cell type that's unique to human and primates. So we're conducting a genetic screen just like what I told you in Perturb-seq. And by collaborating with Aviv and Xiao Wang, a faculty at Broad and MIT Chemistry, we want to use in situ sequencing called StarMap to acquire both the transcriptomic and the information as well as the location of the cells. For example, example, in this very simplified cartoon showing here, you can imagine that there are two orange cells that may be classified transcriptomically as a very similar cell type, but they might be sitting in completely different cellular environments. So by using spatial transcriptomic readout, we want to ask how the mutation affect the gene program may affect the location of these cells reside in, or how the cellular neighborhood can affect the manifestation of the mutation, as well as how the mutation can actually affect their neighboring cells. And hopefully we'll be able to tell you more about this very soon. Lastly, but not the least, I want to thank my mentors, Paula and Feng, without whom I simply would not be here, as well as our collaborator Aviv and Joshua. And I was very fortunate to work with a very creative computational biologist, Sean, as well as two brilliant young undergrad and research assistant, Michelle and Amy, who are now in grad schools. And this project really would not be possible without a strong collaboration with the human geneticists Elise, Ben, and Mark, who taught me how to think about human genetics, as well as all my fundings. Thank you. I'll be happy to take any questions.
Maybe I can step in just briefly. Xin, that was a fantastic talk. Thank you so much for presenting to us. I want to make sure that we are able, we're getting questions on Slack and on Slido, and I think we've had a couple from Slido that I'll pull up right now. First, since the libraries are delivered to early embryos, how do you make your sgRNAs cell-specific? How do you control for off-targets using this method?
So first of all, we do not meant to make our guide RNA cell specific. We actually meant to knock it out in the neural progenitor cells and observe this early deletion effect that may affect the cell type determination and maturation. In terms of the off-target effect, we try to use different guide RNA. We try to validate our guide RNA, but I think in general, just like any genetic screen, the hits are just hits. They are interesting hypotheses that really deserve to use orthogonal experimental system to further understand the mechanism.
Great, thank you. I think we have time for just about one more. Next question is, it looks like you had a relatively large fraction of microglial cells. Was that expected?
Yeah, we did not expect that, and we did a lot of quality control. And it seems like the microglia cells from the single cell data actually express the fluorophore as well as the barcode themselves. So unlikely that microglia cells pick up the barcode indirectly from other cells. How are they labeled? It's likely that the early injection at embryonic day 12 might inject into parenchyma, for example, that induce local injury, and therefore some of the microglia cells might pick up the lentiviral transduction. That's a great question.
Great, thank you so much. I think we will transition to the next talk in the interest of time, but as everyone has been already doing, continue the discussion, please, in our Slack channel. I will turn it back over to Charles to introduce the next talk.
Yes, can you hear me?
Yes.
Okay, thank you very much. Our next speaker is Vijay Sankaran, and he's Chief, Division of Hematology and Oncology, Boston Children's Hospital, an Associate Professor of Pediatrics, Harvard Medical School. Vijay received his PhD and MD degree from Harvard Medical School, and his research group focuses on understanding how genetic variation alters human blood cell production and hematopoiesis. Thank you, Vijay. Please take it away.
Yeah, thank you so much, Charles, and thanks to the organizers for the opportunity to come here and talk a little bit about our work. My goal in the next 15 minutes is going to be to give you an overview of some of the work that we've done from going from maps to mechanisms to medicines in the context of blood disorders. And I hope to briefly talk about a few vignettes and then dive into one recent vignette in more depth. So every second, as each of you is sitting here, you're producing millions of red blood cells, white blood cells, and platelets. And you do this through a well-understood process of blood cell production or hematopoiesis. While we take this process for granted in maintaining our health, it's important to bear in mind that nearly 1/3 of the world population has disorders that impact the process of normal hematopoiesis. And so, for example, I've illustrated here the worldwide prevalence of anemia due to disordered red cell production, where you can see it's widespread throughout the world, with some regions particularly heavily affected. While much of the burden of these diseases can be attributed to nutritional and infectious causes, there's certainly an enormous burden for genetic variation in contributing to these disorders. Our interest in understanding how genetic variation contributes to blood cell production started about 17 years ago, when we were interested in trying to understand how the most common monogenic disorders in the world, those disorders affecting beta hemoglobin, have tremendous clinical variability and diversity. This includes beta thalassemia, that's due to defective production of the adult beta globin molecule, resulting in anemia in patients, and also includes sickle cell disease, which is due to mutations within the beta hemoglobin molecule that cause the hemoglobin to polymerize, deforming red cells and resulting in a number of clinical manifestations. Both of these disorders are known to be ameliorated by increased production of a fetal form of hemoglobin. Now, throughout most of gestation, we express a form of beta globin known as gamma globin, which, when combined with the alpha globin molecule, forms fetal hemoglobin. This is switched off shortly after birth, and then there's increasing production in a reciprocal switch of the adult form of beta globin, shown in purple. This is the gene that's mutated in both sickle cell disease and beta thalassemia, and so continued production of gamma globin can ameliorate symptoms in this disease. It's long been known that this can have a clinical benefit, but how this process was regulated was poorly understood despite decades of study. We were fortunate to be involved in one of the earliest genome-wide association studies to look at variation in fetal hemoglobin levels. And this revealed 3 regions of the genome that were associated with fetal hemoglobin levels, including this region on chromosome 2 within the gene BCL11A that intrigued us. We were able to show, and subsequent work has further characterized, the role of BCL11A as being a critical regulator of the switching process, where when BCL11A is well expressed, such as at the stages of adult development, we know that it silences the gamma globin genes and thus results in predominant production of adult hemoglobin. This is what's occurring in all of our bone marrow right now. And at the earlier stages of development, where BCL11A is not as well expressed, there's increased production of this gamma globin molecule, thus preferentially resulting in fetal hemoglobin production. Now, this work has not only laid an important understanding of how this switching process occurs and given us fundamental insights, but has now led to a variety of ongoing clinical trials to be able to target BCL11A in patients with sickle cell disease as well as thalassemia using gene therapy and gene editing approaches with the attempt to cure these diseases. For example, I'm listing one clinical trial that's being conducted at our center using gene therapy approaches. These early successes in the understanding of fetal hemoglobin regulation have motivated us to ask whether we could further use insights from human genetics to be able to better understand the process of hematopoiesis. Now, certainly we've learned a lot about this process through studies in model organisms, but we would like to try to take advantage of human variation to gain further insights and to try to ask whether we can gain insights into both mechanisms as well as potential medicines through these approaches. Our goal in these studies is not just to conduct the maps, but as with much of the efforts in this consortium, really trying to finish and complete the arc of going to try to understand mechanisms through a variety of approaches, with the goal of ultimately getting at underlying physiologic mechanisms and ultimately understanding how this is perturbed in disease and how we may be able to therapeutically intervene. I want to briefly just talk about a couple of vignettes from our work. So through a wonderful collaboration with Nicole Soranzo and a number of other colleagues, we've been able to now identify thousands of genetic variants associated with blood cell traits. We've been able to also show how these genetic variants then modify the outcome and clinical course in blood diseases. We've been able to map genetic fine mapping data with epigenomic data such as chromatin accessibility to gain insights into relevant cell states and ultimately more holistic mechanisms that underlie various traits. We've also been able to try to, at particular loci, define underlying mechanisms in variant to function arcs and really try to understand some of the underlying biology. And I've just given you one example here. Well, we've certainly learned a lot about normal blood cell production, as well as how this process goes awry in disease, one of the questions we've wondered is how we could start to take some of the approaches we've developed and gain further insights into blood cancers, for instance. And our interest has really been focused on this group of blood cancers known as the myeloproliferative neoplasms. This is a set of disorders that are characterized by excess production of mature myeloid cells. This can include excess production of red cells, excess production of platelets, or of white cells. And all of these disorders can then progress and give rise to acute myeloid leukemia. Now, work over the past 15 years has defined almost the entirety of the somatic mutation landscape underlying and driving these disorders. And this includes the very common JAK2 V617F mutation, as well as a handful of other mutations that explain the vast majority of cases of these disorders. But one feature of these disorders that's been of particular interest to us is the fact that there's a tremendous heritable risk. As illustrated by the Swedish population study, it's known that individuals with a first-degree relative who has an MPN themselves have a 5 to 7-fold increased likelihood of going on to develop a myeloproliferative neoplasm. Some work in the past decade has defined some of the genetic variants underlying this risk. For instance, there's a haplotype of the JAK2 gene that's known to confer some increase in risk, as well as a handful of other variants. However, we sought to try to understand more globally, what are the other variants? And beyond just defining the genetic variants, could we start to gain insights into mechanisms and how the process of normal blood cell production may be altered to give rise to the increased risk of conferring a myeloproliferative neoplasm? The work I'll be discussing was led by Eric Bao, Satish Nandakumar, and Xiaotian Liu in our laboratory. We initially started off by conducting a genome-wide association study of about 3,800 individuals impacted by myeloproliferative neoplasms in a total population of 1.1 million individuals. This resulted in us identifying 17 genome-wide significant associations, as shown here in this Manhattan plot. We initially wanted to understand to what extent do these genetic variants confer risk, and we developed a polygenic risk score of about 100 variants. We could show that this polygenic risk score explained more of the variation than sex, age, or other covariates that we had. And indeed, if one looks at the top decile of risk, as shown on the right, one can see that there's about a 3-fold increased risk for those at the top decile as compared to median deciles, and about a 4-fold increased risk compared to the bottom deciles. We then wanted to go on and try to understand and use in vivo phenotypes to gain further mechanistic insights. All blood cells are produced from a common set of stem and progenitor cells, as illustrated here in simplifying the diagrams I've shown you earlier. We could then look at genetic correlations between myeloproliferative neoplasm risk variants and those conferring variation in different blood cell traits. And across a number of distinct traits spanning various lineages, we could see a positive genetic correlation, suggesting that these risk variants were likely acting at the early stem and progenitor stages to affect the process of blood cell production. We then wanted to get at this using an independent metric. And leukocyte telomere length serves as a nice measure of blood-forming stem cell activity. And therefore, we can use leukocyte telomere length to ask questions about overall blood-forming stem cell activity and variation in this process. At a specific locus, the TERT locus, we can see that there was a positive genetic correlation between variants affecting telomere length and those variants that increased myeloproliferative neoplasm. However, taking this out across the entire genome, then, we can ask across all significant loci, did we see a positive impact when treating telomere length as an instrument in a Mendelian randomization? And indeed, what we could see is that if one treats telomere length as an instrument, we indeed get a positive effect upon the risk of myeloproliferative neoplasms. Once again, nominating stem cell activity and likely driving some of this risk. Now, while these in vivo phenotypes have suggested a role for stem cells, we wanted to see if we could get at this using independent approaches. One approach we could take is to start to ask whether fine-mapped variants overlapped at all with accessible chromatin in specific cell states. And indeed, if we do this analysis, we can see that there's strong enrichment in stem cells as well as multipotential progenitors upstream of all common blood cells, as shown here. If we look across the entire genome and use partitioned LD score regression, we can once again get a strong enrichment in stem cells as well as multipotential progenitors, really illustrating that this compartment is likely to underlie much of the risk. Since I've thus far shown you that we can get at causal variants as well as potential causal cell types, we then wanted to start to gain insights into potential target genes for this variance as well as potential pathways that were impacted. To do this, we could take a number of distinct approaches. We could nominate in a couple of instances where we had a coding variant, the gene that contained a coding variant, and in the majority of cases where we have non-coding variants, we could nominate genes either that were containing that variant within their gene body, or that were connected to a particular gene either through promoter capture high C or RNA-seq ataxi correlations that we could perform across the hematopoietic cell types. Using this approach, at about 70% of loci, we could nominate at least a single causal gene. And I would note that the vast majority of these 15 genes that I'm showing you here are actually factors that are well studied to be important for stem cell self-renewal and function. Indeed, they're highly expressed in the bulk cell populations as shown in the stem cell compartment, and even in single cell data, we can see that they're nicely enriched in the stem cell compartment as well. Now, while these global analyses could certainly nominate stem cells, we wanted to conduct targeted variant to function analyses to provide further insights and really to perturb this process to identify whether this process was impacted. And I'll just give you one example for the sake of time. So we identified a set of variants in a locus about 10 kb downstream of this gene GFI1B. When we then looked for regulatory activity of each of these variants, we found that only a single variant harboring regulatory element seemed to show exogenous activity with reporter assays. And indeed, if we take the risk allele, we saw reduced enhancer activity using an integrating reporter assay as shown here. Moreover, then we decided to go into the endogenous locus and we could perform genome editing to remove the 1 kb regulatory element that I've shown you. And we could do this using 2 independent pairs of guide RNAs. In the bulk population, then, this results in a reduction of about 30% in GFI1B expression. And in a phenotypic population that's enriched for long-term stem cells, we see a similar reduction in GFI1B. I would note that there is no other impact upon any other adjacent genes that we've examined. When we then take these cells out and allow them to expand in culture, we see that there is a more profound expansion in the long-term stem cell compartment as compared to other compartments as shown here upon deletion of this enhancer element. We could then get at this using an independent approach where we could, for example, target either GFI1B in the coding region where we see a reduction in erythroid colonies, but an increased propensity to have secondary replating and self-renewal of these colonies. If we then target the enhancer in contrast, we see no defect in erythroid colony formation, but we see increased production of both primary and secondary colonies as shown here. All of this data and other work that I haven't had a chance to show you has really resulted in a model that we're further pursuing for why individuals might be at variable risk for getting these blood cancers. In that there are individuals who are at lower germline risk and therefore have a certain degree of self-renewal, and individuals at higher germline risk and across the spectrum of variation then have higher degrees of self-renewal, thus increasing the risk of acquiring somatic mutations causing these disorders. So what I hope I've illustrated through the brief vignettes I've talked about, as well as the longer vignette on MPNs today, is really how we can start to develop both maps and at least using more targeted assays, start to try to understand underlying mechanisms and ultimately gain insights into physiology and hopefully inform, as I've illustrated in the case of fetal hemoglobin, therapeutic strategies that could be adopted. Certainly as one challenge that remains is how we can scale up these approaches, particularly as we're targeting primary cells. With that, I just wanted to finish off by acknowledging the people in our group who conducted the work I've discussed today, particularly Eric Bao, Satish Nandakumar, and Xiaotian Liu, a number of other lab members, our collaborators and funding sources. And with that, I'm happy to take any questions. Thank you very much.
Yeah, thank you, Vijay. And I'm in the Sligo, and you're getting some very, very strong compliments about you being a giant in your field. And that is fantastic talk. I think there's a question. I'd say I detect causal variants that show positive correlation between telomere length and MPN risk, coding or not coding.
Yeah, that's a great question. So the variants, and there's actually likely to be multiple independent causal variants at the TERT locus, all appear to be non-coding, and there are at least appear to be at least 2 independent signals in this locus when we've done conditional analyses. So there's a lot more biology, I think, to understand about how this specifically confers risk. Some of this overlaps with variants that are known to confer risk of other cancers, I should say, as well, and are likely pleiotropic, but I think there's much more to really understand about some of the underlying mechanisms.
Thank you very much. And we are going to move in. Thanks, Vijay. We're going to move into the next talk. Again, our next speaker is Naomi Wray, and she's a professor at University of Queensland in Australia. And she's encouraging us to add math into mechanism to medicine maps. Thank you, Naomi, and please take it away.
Well, thank you, Charles, and thank you to the organisers for putting together this fantastic meeting, and also for giving me this time zone friendly time slot, since today's meeting started at 2 a.m. for me. And of course, that's placed me as a statistical geneticist in this session, which is about mechanism to medicine, which in fact I know nothing about either of those. Nonetheless, I'm going to try and rise to the occasion and provide some comment which I hope will be relevant. So I'm going to start with a couple of vignettes. So the first is work by a PhD student in our lab called Yada Wu, who when he first arrived decided that he'd take on the challenge of looking at the medication data within the UK Biobank. So you could ask, well, why study medication? Isn't it just reflecting the disease that people are taking medications for? But I think you can imagine that there are some scenarios where the same medication is given for different disease indications, or similarly, people with the same disease have different medications. And trying to pull those apart could be an important contribution to making progress in common complex disease. So the UK Biobank participants were asked to turn up to an interview, bringing with them information about the medications they took weekly, monthly, or 3 monthly. So this is self-report, but it's not just off the top of your head self-report. This is a prepared self-report, but it is very limited because, of course, we don't know the duration that medication has been taken nor the dosage. So the way the UK Biobank data was collected, that they had 6,745 pre-programmed medication entries, and Yada found that over 1,800 of those were endorsed by at least 10 participants. With his medical training, he then took on the incredible task of manually mapping these to the ATC drug classification codes, identifying active ingredients and doing that mapping. And through that process, he identified 23 medication taking traits. So this is where we have cases, people who take the medication, and controls, people who don't take the medication and don't take any related medications, and took those into genome-wide association study analyses. So this work was published in Nature Communications last year. And although in that paper we highlighted the importance of the work and the key results that we got from the work, what I want to emphasize here in this talk is what we couldn't find, because although the UK Biobank is a wonderful resource, the largest resource we have for this sort of analyses, that the data themselves are limiting. And so this paper is a very nice review talking about drug repurposing in Nature Reviews Drug Discovery last year, and that concludes with 6 recommendations. And one of those recommendations is that the need to move forward with drug repurposing, which of course in the context of common disease might be a way that we could move forward quickly. But one of the first recommendations was this need for big data of data sets with clinical notes, clinical phenotypes linked to omics data. And in many of the large data resources we have at the moment, we are really very limited on medication use traits. So the next vignette I'm going to talk about is thinking about medication in the context of medication, which again is something that occurs very frequently in the context of common disease. So here you can imagine that you have one disease, you take a medication for that, and just by taking that medication, that increases your risk for another disease down the track. Or similarly, common diseases are common. That means people can often have more than one disease. What happens, how do you take, what medication is best prescribed for you for your second disease in the context of taking medication for the first? And these, I think, are very important questions that are facing clinical settings. So I'm going to give a vignette of work by Sonia Shah, a postdoc in our group, who is supervising Salal Shokay, who's a master's PhD student. And the question that Sonia was interested in addressing is to think about the well-known fact that the incidence of cardiovascular disease is 50% higher in those with psychiatric disorders compared to the general population. Now, there's very many hypotheses about this, whether it's cause or consequence. Regardless, we know many people in the population, as they age, take drugs to reduce blood pressure. Are there ways that we can decide which of those drugs are best suited for people who also have psychiatric disorders? So Sonia took a Mendelian randomization approach. I don't have time to go into the details of the pipeline. I'm just going to cut to the quick of the outcome of her work, where passing all sensitivities and multiple testing thresholds, we come to the conclusion that ACE inhibitors, so angiotensin converting enzyme inhibitors, which we know reduce ACE expression, they lower blood pressure, but they also increase risk of schizophrenia. So the purpose of this sort of work is it's relatively cheap, it's relatively quick. It allows us to examine many hypotheses and cut down to ones which should be followed up further. So obviously this is just a statistical result that Sonia's trying to take forward by looking at other healthcare data with the view of are ACE inhibitors bringing on symptoms in those with schizophrenia, bringing on new episodes, or could they actually be bringing on late onset schizophrenia? So with those two vignettes, I now want to turn to some more general thinking of the International Common Disease Alliance, and really to congratulate everybody who was involved in putting together the white paper, and how I fully agree with all the recommendations that are there. There's so much in there and so much nuance. And just to say how important this is for people like me working in a country like Australia, where our budgets are relatively small, that we can write in our grant applications that we're doing things which address recommendations of this alliance. But, and there is a lot of nuance in there and understanding of the complexity. And in all of today's talks, we've really been demonstrated the complexity that we're dealing with common disease. And so a little niggle for me was this very linear suggestion that there's going to be maps to mechanisms to medicines, which really I think we know, doesn't even hold for Mendelian disease. I would note that there was a caveat saying that we don't always think it's going to be linear, but I do think there's a linear vibe to the way it's being presented. And I think the purpose of a white paper is to communicate to people who are outside our field. So I think communicating more complexity would be important. I noticed that Common Disease Alliance has aims to improve prevention, diagnosis and treatment. And so for me, I would have liked more emphasis on prevention and prediction within the white paper. Prediction, because we can go from maps to medicine, totally bypassing mechanism, because the underlying mechanism is irrelevant if the prediction is useful. So the white paper made me look back and think about previous white papers, and so I came to this one from Francis Collins in 2003. And obviously we've made massive progress since then, but many of the challenges remain. And thinking forward from that in 2005, if you think I remember in our field there was just massive excitement as the Wellcome Trust Case Control Consortium put forward the first very large genome-wide association study of 2,000 cases and 3,000 controls. And many people thought that that would demonstrate and identify, the causal variants for common disease. So coming from our background, Peter Vischer, Mike Goddard and myself, we come from a background of quantitative genetics in non-human species, where we've really experienced the power of polygenicity. Our feeling was that common disease was going to be very highly polygenic. And so before that first genome-wide association study, we published a simulation study which looked at this concept of what's now called polygenic risk score. And then went on, of course, to do the first implementation when we got access to genome-wide association study data. So I'd just like to indulge myself to note that one of the things we said in that paper was that the value of these predictive SNPs could be reaped long before the causal mechanism of each contributing variant could be determined. And certainly from today's talks, we can see how much polygenic risk scores have been discussed. And, of the 23 recommendations, only recommendation that talks about things going directly into the clinic is this use of polygenic risk scores. So that's a long-term theme of my research, and I have a recent review, which is a basic primer. And the key purpose of that paper was to make sure we don't oversell what polygenic risk scores can deliver, because of course polygenic risk scores are only able to access the genetic component of risk and also only part of that genetic component. So I know many people might have seen this before, but many people come from different backgrounds. And so I want to just use this visualization to remind ourselves what we mean about a polygenic disease. And so I use this little visualization where I'm imagining a disease which is underpinned by 900 risk loci. There's 900 little dots here. Obviously, we know common disease is underpinned by thousands of risk loci. I've colored the little dots grey if the person has no risk alleles, blue if they have one risk allele, and red if they have two risk alleles. And there's some modeling assumptions in there. And what I'm showing is these risk profiles for cases and for controls. And there's two key things that this visualization shows. First is that there's a higher burden of risk alleles in cases compared to controls. But, every single one of us is walking around with masses of risk variance. It's the only way to understand for polygenic disease to work is for people to have a high burden of these risk alleles, other people that have highest risk. And the other take-home message is to recognize just how different each portfolio is, and the higher the number of risk alleles, everyone will have a unique portfolio. And so that's the reality of what we're dealing with. And so for me, one of the messages I would like to be thinking about is how we take forward paradigms which are not reductionist, but take account of this polygenicity. So I do think there are important questions in the clinic, as I try to talk about, and patient stratification in precision medicine is something we discuss, but how do we get the data to make that stratification? So we're going to need more data in order to pull apart these different scenarios. And what I think we need to aim for as we go forward is to embrace this variation, is to think about how we can make molecular paradigms which aren't reductionist, but use this variation. So we've heard a lot about ways in which new technology is coming to mechanism. And I think those new technologies can be used to understand these vulnerable genomes. So what I'm trying to say is that in the white paper, we talk about using biobanks and making sure that there's biological samples. I think it's also important to push that those biological samples can be used to generate cell lines, and that those cell lines could then be used to generate essentially cellular biomarkers. So the white paper already talks about collection of biomarkers, but that's really talking about biomarkers collected from the standing person. I'm talking about collecting biomarkers which are derived from those cellular phenotypes. So we can use the, let's say, the advances that are being put forward for mechanism, but using them to understand variation between people. So take people who are high risk, have vulnerable genomes, recognizing that every single one of them is going to have a unique portfolio, compare those to resilient genomes, and generate biomarkers which can be put together with the clinical phenotypes into machine learning type algorithms to improve predictors, and therefore understand why some people respond to treatment and others don't. So I realize that this is futuristic in that these technologies at the moment aren't yet scaled up, but I think that these are the paradigms that we need to be thinking about, how we do scale them up and how we prepare for tomorrow, how we make sure that we have the biological samples which could benefit from these technologies. So in summary, I've shown how I think medication use should be used as a trait in our analyses and how that can contribute to the maps. That in common disease, we know that medication use in the context of medication use is something that clinicians are crying out for guidance on, and we need more data to be able to contribute to that. I've championed the importance of the white paper, but made a small criticism in the way the non-expert reader is positioned. And I'm suggesting that version 2 of the white paper would better position the non-expert reader to embrace non-linearity, embrace the complexity. And I would like to see an increased profile on prevention and prediction. I would like to promote the collection of biological samples that can be used to generate cell-based biomarkers and that use those in prediction and patient stratification paradigms, because I do think that these will be able to deliver more quickly than many of the ways in which the progress from maps to mechanisms to medicines. And so I came up with my own little meme, and I'd like to end with acknowledgements to the work of the people that I've talked about, and of course the funding agencies. Thank you.
Yeah, thanks, Naomi. Excellent talk. Again, I'm just going to start with my own question. I was wondering, how do you model a trait like hypertension, or you predict it, given that over 90% of us will get hypertension if we live long enough?
Well, I'm saying that, you know, I think there are for diseases where many people get the disease, and then there's also age of onset comes into that. And what we know is those with the highest polygenic risk tend to have earlier age of onset. So I would still be, I'm not even proposing that you're directly modeling disease, but rather take the genomes of people that you know are at high risk, take the genomes of people you know are at low risk, and rather than do things like making isogenic cell lines and introducing perturbations, I'm saying use the natural variation that's there to see if we can generate cellular biomarkers which can be used in predictive settings.
Thank you. We have a question. It said, as public large data sets and machine learning methods become more widely available, how do we encourage study design utilizing best, in brackets, statistical and ethical practices rather than the path of least resistance approach?
Well, there's many questions in there, some of which are not part of my expertise, but in terms of the methodology, you know, I think many people are very interested in using machine learning, and often we're trying to apply it to data sets which just aren't actually up to, you know, what the machine learning methods could deliver. So I'm saying let's try and generate the data sets which would allow us to apply machine learning in order to make those predictions. In terms of the ethics and the policy, I think you know, that's obviously really important, but I'm not really the one well placed to respond to that.
Thank you. I also wanted to encourage the audience to remember that the next panel will also be talking about similar issues of mechanisms, and if you have questions, to also pose it you know, to that panel, and there will be opportunity to answer your questions. Again, you know, thank you, Naomi and Rachel.
Great. Yes. Thank you so much to you, Charles, and to all the presenters for a fantastic and really an energizing session, I think late in the day for a lot of us. And it was really great to see so much energy and enthusiasm for this work.